I'm Genta Nagai from the University of Tokyo, and it's a very big pleasure to be here in this meeting. So today's my topic is about direct detection of DNA modification in cancer genome. So first topic is about DNA methylation in allele-specific manner in cancer genome. So as we know everybody, so DNA modification is a crucial modification for the epigenetic regulation in human cells. And this modification added by the DNMT, DNA methyltransferase, and also erased by during the DNA demethylation pathway by TET family proteins. In this cellular differentiation, in young cells, the promoter region unmethylated states, but the other gene body regions are methylated. But during the aging or cancer development, some tumor suppressor gene or cancer-related gene show promoter methylation in cancer genome. So recently, the improved accuracy of base and modification calling and recent chemistry achieved greater than Q20 accuracy of the base calling and also the methylation calling. So it's time to use for cancer genome sequencing. So we prepared the DNA, long DNA, genomic DNA, using the Purigene kit by QIAGEN, and also fragmented the long genomic DNA up to 10 to 20 kb in length, and also sequenced by the P24. So that is a typical example of the basic statics of the read length, and the read length is about 10 to 20 kb, and we sequenced gastric cancer organoid genome up to about 30x. And then base calling by GUPPY, and also mapped to the human reference genome. Okay? So that is an example of the aberrantly hypermethylated and hypomethylated region. MAC5AC is a well-expressed gene in enterocyte, normal gastric site. So this promoter region is blue color, and also this is densely methylated in gastric cancer organoids. Vice versa, the one-cut gene, HNF6, is aberrantly activated in the cancer cell. So this region is aberrantly demethylated in promoter region. So there are the unique genes in with a differential methylation in allele-specific manner. So there is some kind of the gene set that is called imprinting control region in mammalian genome. So as shown here, the KCNQ1 OT1 is methylated in paternal allele, but methylated in maternal allele. So that is a differential methylation pattern in imprinting locus. So we use the nanopore sequences that show that a distinctive pattern of methylated color and unmethylated color, like a PEG10 or KCNQ1 region. That is a well-known imprinting region. So we can see clearly show that distinctive pattern of red color and blue color. So red color represents methylated allele, and blue color means unmethylated allele. So that is a very simple locus of imprinting control region, but in that genus locus, there are so many imprinting regions. So this is very difficult to analyze by the short-read sequencing. So nanopore sequence is a very strong approach to elucidate that complicated imprinting center. So we also checked the all types in promoter region of the IGF2 gene in adult liver. So this blue color is the promoter 1. This is unmethylated state, but red color is called as fetal promoter. This is a 50% of methylation by whole genome bisurface sequencing. In nanopore data, we can clearly show that the clear pattern of imprinting methylation only in fetal promoters. So that's a powerful tool to detect or accurate calling of imprinting methylation status. So we also analyzed the allele-specific methylation in mesothelioma cancer cell lines. So we also found that de novo allele-specific methylation in cancer cells. So in this HOXD cluster region, we can see that most of promoter region are unmethylated in normal blood samples, but in tumor, densely methylated, often observed. So in this HOXD8 region, we can clearly see that aberrant methylation in tumor cell, but we couple with the haplotyping, we can see that dense methylation only in haplotype 1. And also analyzing with RNA sequencing and chip sequencing, the only haplotype 2 show RNA expression and also active histone mark. So this region is differently regulated in cancer genome. That is other example of the antisense gene in HOXD region. So we found 5,000 of allele-specific de novo acquired methylation in mesothelioma cell lines. Okay. And another topic is about hydroxymethylation. This is a very important modification to consider the methylation dynamics in human genome. So in this DNA demethylation pathway, TET family gene convert 5-MC to 5-HMC. And this intermediate metabolite is deposited in gene body region of expressed gene or CPG iron shore region by the previous report. So we checked that modification in cancer genome. So this is a typical view of the GAPI modification core of the human gene, and also ApoE gene is well expressed in the gastric cancer cell lines, and blue color is unmethylated and red color is unmethylated. But with all cytosine modification mode, we can see pink-colored hydroxymethylation in this read information. So we can clearly see that the HMC signal as pink color in the gene body of ApoE region. We can also show that CPD shore region as 5-HMC enriched region. So it's in the core promoter region, MLH1, the blue color that shows the unmethylated status, and in the intronic region, red color deposits that represent 5-MC. And here is the boundary region A and B, and we can show that HMC signal in this boundary region in one base resolution. So that's suggesting that defense mechanism against CPGR and promoter methylation in human genome. So we also studied the aberrant expression of the TET1 upregulated liver cancer, and we previously reported that the ectopic HMC deposition by using 5-HMC deep sequencing is an immunoprecipitation-based approach, and we analyze these identical genome by using nanopore sequencing. This is an example of the TET1 enriched region. So here is the intronic region TET1, and that pink peak is hydroxymethylation signal detected by HM deep sequencing, and we sequence this genome by nanopore. So we can see that the ectopic 5-HMC signal in this region by nanopore sequencing in one base resolution. So another example is the ectopic activation of the oncogene HMGA2, and we found that that enhancer signal is very critical for the gene regulation by chromatin looping between promoter and enhancer region. And we can also see that the 5-HMC signal in this ectopic enhancer region. So this 5-HMC detection approach is very crucial, very useful for the de novo activation in cancer genome. So that is a global distribution of the cytosine methylation and 5-HMC comparing with the immunoprecipitation approach. And 5-MC distributions show a similar result, but 5-HMC have some background in this data I show. So we would like to improve the 5-MC calling algorithm using the latest Dorado coder. So this is my take-home message. So nanopore sequencing provides a long-range landscape of 5-MC and 5-HMC in cancer genome, and more high-depth reads are necessary for quantitative evaluation. So thank you very much, and I would like to acknowledge for our collaborator. Thank you very much.